If I want to describe anatomy, this is coming to you guys. The histology that we will begin, y'all have already started it in lab, have you not? Yeah. So the histology that's coming in this class, you're going to learn that most of the time when we describe things anatomically, we're talking about the nature of the proteins that are there. What's the structure of them? So here's the end of the story. What do you mean, genes? What do you mean that genes run strong in my family? Well, when you get right down to it, what I'm really saying is the sequence on his DNA that gave his cells instructions for how to build the proteins that constructed him are the same genes that I have on my DNA that gave instructions to the cell for how to build those proteins. That's central dogma. If you're talking about genes, you're talking about instructions for building proteins inherent in the DNA molecule. So I'm going to give you a little more language on that as we proceed through this. I have a couple more pictures to look at here. There's the repeating sugar phosphate backbone. Here are the rungs of the ladders with cytosine, base pairing with guanine, and adenine with thymine. So the base structure of the DNA is very simple. The complexity is in the protein. All right, so tell me this then. How does the DNA actually give instructions for making protein? Now, at this point I told you, I asked you to do some reading, did I not? Okay, so it's been a while since you've had this stuff. I understand the language is going to get a little sticky here. So if you've done your reading, keep up. If not, slow me down to talk about this, because I am not going through all the central dogma, but just enough of it so the student has a grasp about what you mean when you say making a protein. Okay, so the process of DNA being used to build a protein can be fit into two simple steps. Step one is transcription, and step two is translation. So in the first step, in the transcription step, what happens is an enzyme causes the DNA molecule to unwind. By unwinding, I mean that one side of the DNA is going to be separated from its counterpart. So these bonds between these base pairs are broken. And one piece, one short segment of the DNA separates itself from each other because of the activity of enzymes within the cell. One side of that DNA molecule becomes what we call the coding strand, and the other side becomes the non-coding strand. So on this picture, when you look at it, this is labeled the DNA coding strand. So what I essentially did was, in this image, uncoil this molecule, cut this part out right along the hydrogen bonds, and flip it over. So these base pairs, T, A, C, 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 would fit here if it was in the right order. T, A, C, C. Flip the thing over. The coding strand is the side of the DNA that is going to be copied. So when you say transcription in language, what you mean is you are making a copy. And that's the same thing in biology. If you are transcribing the DNA, you're making a copy. Here's the difference. The copy made in biology is not a letter-for-letter copy. Instead, it's a coded copy. Because every time there's a T on the DNA, the cell copies A. Now, can you explain that? It's the same explanation I gave just a moment ago for the replication process. If you want to build a new cell, you have to separate the DNA and copy it. Is it copied letter for letter? Does T go with T? No, T goes with A, and C goes with G. Same thing works here. The copy that is made of that piece of the DNA is a semi-conservative copy. But in the case of RNA, there are no thymine nucleotides for RNA. They're uracil. So you can always know when you're looking at RNA, you find U's in there. So everywhere we have a T on the DNA strand, we have an A. Everywhere we have an A on the DNA strand, we have a U. Everywhere we have a C, we have a G. And we make a copy. This is a transcribed copy of the coding strand of the DNA. So step one is unwind a piece of the DNA and copy one side of it. Transcribe it. That copy is called messenger RNA. And the reason it's called messenger RNA is because it now contains the instructions. It becomes a messenger to tell the organelles in the cytoplasm how to build the protein. It is truly a messenger. So the messenger RNA now leaves the nucleus and goes out into the cytoplasm and is going to give the cell instructions. Now, as it turns out, the organelle that is going to read, it's transcribed message, right? The organelle that's going to read the message is not going to transcribe that message. The organelle is going to take the message now and translate it into something completely different. So the process of translation is taking the message on the RNA and converting that message into the language of amino acids. We have now moved from nucleic acid to protein. It is happening now because the messenger is giving the information to an organelle that will now translate it into a brand new language. And this language is the language of the amino acids. And it is very predictable. In fact, there are only 20 amino acids that are used in the human body to make proteins. All of them have codes. I know that if the DNA base sequence is TAC, that the mRNA message transcribed will be AUG. And I know if the AUG message is on the RNA, the amino acid will be methionine. The code in biology for methionine is AUG. That means methionine. It is a coded language that exists inside human body cells. AUG puts methionine. And so all the rest of the amino acids, they all have codes. In fact, most of them have more than one code. So you have multiple codes that give information to the cell. This is a translation process. Turn it from a nucleic acid now into a protein. It's a completely different language. It's no longer base pairs, it's amino acids. It is a language. Now, before I leave this slide, let me say one more thing. If something happens in the replication process, building new cells, or if something happens in the nucleus so that one of the nucleotides that gets added or more is the wrong one, that is called a genetic mutation. And it can have catastrophic consequences. Cystic fibrosis, sickle cell anemia, various versions of other neurological disorders, and many, many others that have been discovered over the last 15 years because of the Human Genome Project, tell us that it's very important. The letters in this gene, the letters in this transcript, and the letters in the code are an extremely important language. They matter to the cell. I told you during the last class meeting, a single base substitution at the 6th position in the beta chains of the hemoglobin quaternary protein is the answer to why we have sickle cell anemia in the human population. Very similar point mutation that occurs in cystic fibrosis. A single point mutation there causes the protein that's made to not transport chloride ions properly in the cell membranes of the pleura. And it causes that mucus buildup. We know what the problem is. This point mutation has been identified. It's another matter to fix it. But we know what the root cause is. The point here is to say it matters. It's extremely important, the order of the letters. And I keep using the word language, and I'm doing that on purpose. Because this is a language that all of the cells in your body, except the red blood cells because they don't have a nucleus, but every other cell in your body, the 300 trillion or so cells, have this language in them. And the sophistication of regulating this language is the sophistication of life. Now, a very famous guy by the name of Francis Collins, he is the current head of the National Institutes of Health. He's a geneticist, and he was in charge of the government side in creating, in performing the experiments that had to be done to finally decipher the human genome. This was finished back around the time many of you were born, around the year 2000. The human genome was a joint public and private venture. It was a multi-million-dollar project to sequence the human genome. And Collins, who's now the head of the NIH, he's arguably the most famous geneticist in the world, certainly in this country. Everybody knows his name. Stood on the platform with the president, announcing the completion of the project. And he said, "Mr. President, we have discovered the language of God." And, the expectation was, we have discovered, the sequence of the DNA in the Human Genome Project is done, and you should give me a raise. That's not what he said. And then he wrote a book by that name. And so I want to show you why he said that. Why did Collins call the Human Genome Project, which was the process of describing the DNA base pairs in the entire human genome? It's a massive undertaking. This is why he said it. Do it with this one. The backbone of the DNA is a predictable repeating sequence of phosphates and sugars. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So there's nothing inherently interesting here. The DNA molecule is interesting in the fact that these letters in threes, CGT or GTC, GTG, the three-letter sequences here that are in the DNA will ultimately be the message that tells us what amino acid gets added to the protein. So the real interest in this molecule in terms of life is this bond. Because it matters that that is a cytosine. That cannot be a guanine or an adenine or thymine. This has to be a cytosine. If it's not a cytosine, that is a point mutation. And that can cause massive problems in human or any other animal anatomy and physiology. The letters matter where they go. Just like if I was going to write some English language out for you. So I'll write the language out, and I put letters in the wrong place, it causes problems in communication. You may be able to understand it, but it's going to cause issues. In the human body, it causes, in many cases, it causes massive issues. So that bond right there is the one that's interesting. If you are going to explain how these letters got in this order, then you have to be able to, and you want to do it, if you want to explain that in terms of natural processes. That is, there is a chemistry here in this bond that dictates the cytosine goes there. Well, now you have a reasonable explanation for randomness. And the neo-Darwinists in our country, and otherwise, especially in this country, in pockets university pockets all over the place, were extremely excited about this process of describing the human genome. Because if you can explain the order of the letters by way of a natural process, then you can explain mutations as a way of changing living things. But here's the problem. This bond here, called the glycosidic bond, doesn't care whether it bonds cytosine or guanine or thymine or adenine. There is no specificity in the glycosidic bond.